Hello dear students, let us understand one more topic, that is RNA processing. In this particular topic we will understand the 5' capping of the mRNA, how it is added, then how the poly A tail is added in the mRNA, and then how the introns are spliced out and exons joined together to form a mature mRNA. So this slide shows the overall overview of the RNA processing. As you can see, this is the DNA strand in blue line. This is the newly synthesized mRNA after the transcription. This, as soon as the 5' end of the mRNA synthesizes, 5' cap is added on this region. Then introns are also started removing during the transcription. So splicing can occur during the transcription, and after the transcription is completed, the poly A tail is added. So these three steps together constitute the RNA processing. So this slide shows the 5' capping, how it is done. So this is the mRNA, newly synthesized mRNA, and this is the first base of the mRNA, this is the second base, and next bases are in this direction. So as you can see, the 5' carbon of the ribose sugar of the first nucleotide of the mRNA is shown here. This cap is added during the 5' capping. This cap is mainly 7-methyl guanosine. 7-methyl guanosine is added to this mRNA via 5' carbon of the ribose sugar of this guanosine, and a triphosphate linkage is formed. So a bond between the 5' carbon and 5' carbon. So in reverse orientation, a bond is formed, and this bond is known as 5' triphosphate linkage. This cap is added by the enzyme called guanyl transferase, and after addition of this cap, few of the first few base ribose sugar are methylated, and this constitute the 5' capping of the mRNA. In this picture, the detail of the 5' cap addition is shown. Different enzymes are involved in the addition of the 5' cap, 7-methyl guanosine. During transcription, as soon as the mRNA synthesizes this 5' region, the cap synthesizing complex attached to the 5' end of the mRNA, and cap is added. So capping is basically co-transcriptional. That means transcription and capping to occur together. This cap helps the mRNA to stabilize it. This cap is also essential for the splicing of the mRNA. That means removal of the intron and joining of the exon. Then mRNA is also transported from the nucleus towards the cytoplasm, and that cap plays important role during transport, and this cap is also helpful during translation. So these are the main function of the mRNA 5' cap. So the second modification that occur in the mRNA is addition of the poly A tail. So in newly synthesized mRNA, these three sequences constitute the signal for the poly A tail. This is known as polyadenylation signal, the first signal. This second is known as cleavage site, and this is the last part of the newly synthesized mRNA, which is rich in G and U. So after cleavage of this cleavage site, a tail of adenine residue is added, and this modification is known as poly A tail or polyadenylation of the mRNA. So how this is done? First, the cleavage at the CA site is occurred by the endonuclease enzyme. Then string of adenine nucleotide is added by the polymerase enzyme. So this addition of poly A tail help the mRNA in increasing its stability and thereby increase the protein concentration that is coded by that particular mRNA. And this poly A tail also help the mRNA during translation. So these are the different associated proteins that are required for the addition of the poly A tail. As you can see here, the first is CPSF, that is cleavage and polyadenylation specific factor. This factor is essential for the recognition of the poly A signal. Then cleavage stimulatory factor. Cleavage factors, there are many cleavage factor 1, 2, 3. Then poly A polymerase and enzyme and poly A binding proteins. They bind to the poly A tail and stabilize the poly A tail. So this CPSF protein first recognize this poly A signal and bind to the poly A signal. Then cleavage factors and stimulatory factors then bind, and then they cut the cleavage site, and polymerase enzyme synthesize the adenine residue at the last tail of the mRNA, and this tail is stabilized by the poly A binding protein. So next is RNA splicing. So presence of intron and exon were first discovered by the Philip Sharp and Richard Robert. Exons are the coding region of the mRNA and introns are non-coding region of the mRNA. During splicing, these introns are spliced out and exons join together to form a mature mRNA. And this mature mRNA, after splicing, transport out of the nucleus into the cytoplasm, and then translation began. So there are different groups of the intron. How this splicing occur? Based on that, there are 4 main group of the introns. So let us understand splicing in different groups one by one. So in group 1, the splicing occur by an external guanosine. So this is the exon, and this yellow line is intron, and this is the next exon. So in this particular group 1, external guanosine act as a nucleophile. The 3' hydroxyl group of the guanosine act as a nucleophile and attack the phosphate at the 5' split site, and this guanosine then bind to the intron. So this guanosine is now bind to the intron. Then the 3' hydroxyl group of this exon then act as a nucleophile and attack the phosphate group at the next exon, and these 2 exons join together, and this intron is released in a linear form, and the guanosine is now a part of the released intron. So this is the group 1 intron. Then group 2 intron. In this group 2 intron, the hydroxyl group that makes nucleophilic attack is a part of the intron. So second hydroxyl group of the adenine residue in the intron itself, this is known as branch point sequence. So this hydroxyl group act as a nucleophile and attack the bond in front of the 5' G of the intron and create a lariat, and that lariat is then free out of that mRNA, and these 2 exons then join together. So this is one more slide of group 2 introns. So how the 2' hydroxyl group of the adenine of the branch site makes an attack first at the 5' side of the exon. Then this free exon then makes the second attack, nucleophilic attack, and these 2 exons join together, and this intron is released as in form of lariat. So then group 3rd intron. So in group 3rd intron, we have many different proteins involved in this splicing reaction. First, U1 snRNP and U2 snRNP, they bind to the 5' side and 3' side or branch site of the intron. Then U4 U6 complex, then U5, they bind to this intron complex and make an inactive spliceosome. Then ATP is utilized and activate this spliceosome, and U1 and U4 are released. U5, U6 and U2, they make an internal arrangement and thereby release the intron in form of lariat, and 2 exons are joined together. So in the group 3rd intron, U1 and U2 snRNP are the first ribonucleoproteins that recognize the intron and bind to the introns. Then remaining snRNPs U4, U6 complex and U5 bind. Then internal arrangement convert the mRNA in such a way that introns is released as a lariat and two exons joined together. So in group third intron, as you can see, involvement of the ribonucleoprotein in form of U1, U2, U4, U6 and U5 occur, and they are essential for the splicing reaction. In group 2 intron, the hydroxyl group of the intron itself was acting as a nucleophile, but in group 3 intron, external RNA comes here and play a role of nucleophile group and make internal arrangement in the intron, and intron is released as a lariat form. So this slide shows the different groups of the group third intron. So the three main class of the group third intron. Class first is known as major class because the protein are U1, U2, U4, U6 and U5 as we have seen in the previous slide. Then class 2 is U12 type because in this particular class, instead of U2 group in the branch point, we have another snRNP that is known as U12 snRNP. So U11 and U12 are involved in place of U1 and U2 in this particular splicing reaction. So they have a different recognition sequence. As you can see here, the sequence is GU, while U11 recognize UAU sequence. So there are about 0.1% introns that contain this sequence in the human, and that is recognized by the U12 type of the spliceosome machinery. The third class is known as trans splicing. In trans splicing, as you can see, the snRNP is consumed during the reaction, and part of this snRNA becomes the first exon of the mature mRNA. So this snRNP becomes the part of the mature mRNA. So that is why this is known as trans splicing class. So there are three classes within the group third intron: major, U12 and trans. Then last group is group 4 intron. This is mainly found in the tRNA, and the reaction is almost same as we have seen in the group 1 and group 2 intron, but these type of groups require the energy in form of ATP, and endonuclease enzyme is involved for the splicing out of the introns, and in this endonuclease cleave both end of the intron, and then exons are joined together by the DNA ligase enzyme. So we can see that there are 4 different groups of the introns, and out of these 4 group 3rd intron have 3 more class: major, U12 and trans type. So these are the scientists who were given the Nobel Prize for their discovery of the RNA having catalytic property. So this is all about. In this particular video we have understood about the 5' capping, addition of the poly A tail and splicing